Hello, my name is Gisele Vajgel. I'm a nephrologist here in the Hospital das Clínicas of the Federal University of Pernambuco. And on behalf of my co-authors, I'm going to present our study that was recently published in the Journal of Rheumatology. And the title of the study is Effect of a Single APOL1 Gene Nephropathy Variant on the Risk of Advanced Lupus Nephritis in Brazilians.
Since 2010, it's well known that end-stage renal disease is more prevalent among African Americans due to the presence of APOL1 renal risk variants. However, the impact of those variants in mixed ancestry population is unclear. The Brazilians are the mixed population with different proportions of Amerindian, African, and European ancestry. In Brazil, we have few studies that genotyped APOL1. Lupus nephritis is the main reason for non-diabetic kidney biopsy according to the Pernambuco Registry of Glomerular Diseases. Pernambuco is our state in northeastern Brazil. The primary hypothesis of our study was to determine whether there was an association between APOL1 renal risk variants and the development of progressive CKD defined as a sustained eGFR of less than 30. Secondary analysis assessed the impact of APOL1 renal risk variants on additional kidney outcomes in lupus nephritis, including kidney histology and long-term kidney function. Cases with lupus nephritis were enrolled from 3 outpatient clinics in Brazil, specializing in the treatment of glomerulonephritis. 2 of them were in Recife, northeastern Brazil, including Hospital das Clínicas, in the Federal University of Pernambuco, where we are right now, and 1 in São Paulo, southeastern Brazil. We genotyped APOL1 G1 and G2 risk alleles in 201 non-white Brazilians with lupus nephritis, and in 222 healthy blood donors. We did not include patients with non-lupus nephritis histologic patterns, including those with collapsing glomerulopathy. Genomic DNA was isolated from whole blood and shipped to Wake Forest School of Medicine for APOL1 genotyping using TACMA assays. There were no different frequencies of APOL1 allele between lupus nephritis cases and controls. Because only 4 cases with lupus nephritis had 2 risk alleles, the groups were analyzed based on the presence of G1 or G2 alleles. The groups had similar baseline clinical and demographic characteristics, eGFR, proteinuria, and histologic classes of lupus nephritis. However, we noticed that the initial biopsy from patients with 1 or more renal risk variants had more chronic damage compared to those with 0. They had more glomerular sclerosis, tubular atrophy, interstitial fibrosis, and also higher chronicity index. In this table, we can see that those with 1 or more renal risk variants had more CKD stage 4 and stage 5, and also the time from the initial diagnosis of lupus nephritis to end-stage renal disease was significantly shorter in lupus nephritis cases with 1 or more risk alleles compared to those with 0. Here we can see the Kaplan-Meier renal curves for CKD and end-stage renal diseases with significant p-values. This table displays the outcomes in the 4 lupus nephritis cases with 2 APOL1 risk alleles. Despite this small sample, half of them progressed to CKD stage 4, and 1 has persistent proteinuria after 3 rounds of induction therapy. The low frequency of APOL1 2 risk allele carriers in our Brazilian lupus cohort did not permit performance of outcome analysis using the traditional autosomal recessive model. However, the presence of even 1 allele demonstrated a significant association with advanced CKD during follow-up. Moreover, this dose-dependent cytotoxicity has been shown by other authors, including in vitro studies. It's important to mention that the presence of the genetic variants per se does not preclude kidney disease. A second hit or another insult should be there to trigger the disease and the risk for CKD. HIV and lupus are strong insults as these are conditions that produce high loads of interferon, which stimulate the cellular expression of APOL1. We conclude that frequencies of APOL1 renal risk variants in non-white Brazilians with lupus nephritis are not significantly different from those in healthy non-white Brazilians. But participants with one or more APOL1 renal risk variants had more severe kidney disease at presentation and higher stages of CKD after therapy compared to those with zero APOL1 renal risk variants. And finally, I'd like to thank my co-authors for such a great help and for this opportunity.